How about now? Oh, look at that. No echoes at all. Not even, okay. Fantastic news. So so yeah, I know how to read science papers. I think mushrooms are neat. So I've read a lot of science papers about psilocybin mushrooms. And the first thing I want to say is, what kind of mushrooms are we talking about? For the purpose of this conversation, I want to define it as psilocybin containing mushrooms. I mean, you could say psychedelic mushrooms or entheogenic or magic mushrooms, but I want to specifically talk about mushrooms that contain psilocybin, which there are a lot of. A lot of people tend to think of Psilocybe cubensis as the only one, and that is the most popular one, but there are a lot of others, and they're being discovered more every day. So this is what I'm talking about. And the other mushrooms that are quote unquote magic mushrooms or have psychotropic properties, are bioactive, they use different chemicals. So Amanita muscaria is a different biochemical pathway to have its mind altering effects than psilocybin mushrooms. So we're just talking about psilocybin mushrooms, and I want to make a quick note on a couple of these words. These are two words that are commonly used. You can see they're from very similar Greek roots. There's other versions of this, of different people in the space have put together some words and proposed a new word. Psychedelic is the older and the more classic. It's more used in scientific studies, though of course there's lots of other words like psychotropic. Entheogenic is a newer, and there are connotations to these words. So I use them interchangeably, but I do prefer entheogenic. Psychedelic has a little baggage. It has some baggage that it's from this kind of like, let's do everything in the lab, medicalized version of, approach to these substances and these mushrooms. And it has baggage of the war on drugs and, things like that. So entheogenic is kind of entheogenic in my mind encompasses a little bit more of the mystical and spiritual than psychedelic, which can be just very dry and straightforward. Okay. So these are the things we're going to talk about. I want to give a little context first, and then go through the chemistry of these molecules, recent research, and then a little bit of application. And so let's talk about humans and entheogenic examples. Has anyone ever heard this thing about Santa Claus and amanitas and the witch's salve? Are people familiar with this concept? So these are kind of like cultural remnants that echo in our memory, right? And that they give a clue that there was some relationship between humans and mind-altering plants and funguses, but they're pretty vague and we'll never really know, right? And then there's things like soma. Are people familiar with that? That is an ancient Indian thing that there is documented writings about its use and the fact that it was mind-altering and this and that, but we have not been able to figure out what plant it is, right? So there's something where, okay, we know that humans had an interaction with this. And in general, humans have had interactions with entheogenic plants since there's been humans, right? It's only the last 100 years that we think food comes from the grocery store and medicine comes from like a global pharmaceutical company. So we have relationships with these plants. That's all the point of this slide in this moment, is to say, it's not that psilocybin mushrooms are some exception. Like humans have had these kind of interactions with these kind of plants and fungi all over the world for thousands of years. In another category in my mind would be like the toads that you lick in South America, or peyote, where they have been doing it for a long time and they're still doing it. And you can kind of see this unbroken relationship relatively. Buti is a substance in an African country that is a psychedelic substance used in ritual religious purposes, and it's one of the 3 official religions of the country. And I love this idea that it's just kind of, it's just there's 3 official religions and one of them happens to have a psychedelic ritual and it's no problem, right? So that's just to give some context. And the place I really want to focus on history, though, is Mexico. So Mexico is interesting in that psilocybin mushrooms are the substance that we're talking about, the specific entheogen, and also the chain, in my mind, is very interesting. So first you have a lot of art that depicts mushrooms, paintings, glyphs. They have there's these symbolic things. The words for these mushrooms sometimes translated as things like fruit of the gods. And they have specifically these statues that archaeologists and historians believe were for grinding the sacred mushroom, kind of like an inverted mortar and pestle. And that's a really strong sign, that and all the representations of mushrooms in religious and spiritual imagery surrounding it would be a really strong sign. But we don't just have the art that we're trying to interpret. We also have European records. So when the Europeans came, they were very interested in any religious practices and kind of wiping them out, or, you know, wrapping them up in their religion. So there's a lot of records specifically about eating these mushrooms, how they were prepared, the kind of states that you went into, the cultural significance of it. And then the next link in the chain is Maria Sabina. Is everyone familiar with Maria Sabina? Okay, well, I'll still say a few words about her. This is a picture with one of the Watsons right there. And this is a person who had an almost unbroken connection to this. And then she touches on to kind of like the modern American, the modern European and American life. When this person is part of a couple, Valentina and Gordon Watson, they sought her out, hearing rumors of this unbroken tradition of using these plants as medicine, these mushrooms as medicine. And then went on to write a Life magazine article about it and publish some other things that were very popular in the United States. And in many ways, this is an inflection point to the kind of like the lab era is what I'll call it, right? Like, so we have the touching this indigenous tradition to, and then we're going to go into, like I said, what I would call the lab era. Valentina and Gordon Wasson mentioned them. Think about, so this is in the mid 20th century. This is like 1950s, right? And I call it this, I say lab era or medicalized or something. Think about what's going on in the 1950s in our culture. We're damming rivers so we can electrify the West, right? We're moving into like modular little homes disconnected from community. We're shopping in grocery stores, and like gardens and like farm food is for like unsophisticated people or whatever, right? You know, so this is, our approach to this is very different than I think it would be maybe today, or that I would necessarily choose. It's not approach to the sacred medicine or this cultural treasure from another culture. It's kind of approach to like, how can we break it down and reproduce it and control it, right? And that was kind of the approach. And that's the 1940s and 50s, maybe early in the 60s. And then you get into kind of the political campaigns against psychedelics, right? This is when, who can comment on the motivations of politicians, but many legislations and what would I say, like marketing campaigns come out of this era that drugs are evil, right? If you drugs, not meaning caffeine or anything from pharmaceuticals, but meaning these type of mind-altering plants and funguses. If you smoke weed, you'll have reefer madness and have psychosis, right? If you smoke weed, you're gonna put your baby in the oven. So there's, I don't know, that was like a thing in the 80s or something. You know, your brain on drugs with the egg. And also horror stories about people who had made these, you know, they smoked a little bit of weed and just turned into monsters. So that is definitely an era. And in that era, all research kind of shuts down, right? It's just like, and it's kind of almost erased in many ways. Like, it's not even like we used to do that and here's what we learned. It's just shunned as a knowledge base. But then somehow, I don't know exactly how, that mentality, that kind of like war on specific natural compounds starts to fade and research returns. And then you could say the second wave of psychedelic research is one way you could talk about the things that came out. So now let's go into chemistry. And I just wanted to make a quick note that we're not going to talk about specific studies in terms of fundamental research or even very much in preclinical, but most of the things we know is relatively new. We know a lot more about the fundamental understanding of chemistry of these compounds since we started researching again, since the war on drugs has faded. These are the compounds we're talking about, psilocybin and psilocin. Psilocin is the active compound. So we talk about psilocybin mushrooms and we measure psilocybin and things like that. But the thing that actually is interacting with you is psilocin. And there are enzymes in your body that will take that phosphate group off. And then you have the bioactive compound. I'm sorry, can you hear me when I look at the screen? That's a bad habit, eh? There's a family of compounds. There's a lot of, like serotonin is shaped very much like this. Serotonin's in the same family. Tryptamines is one word you could use to describe them, from an amino acid called tryptophan. Do you remember when there was a theory that the reason you were so tired and sleepy after feasting on Thanksgiving was the turkey? Yeah, like that's that molecule. Serotonin, things that make you feel good, mood regulators, right? their roles are really not understood. There a lot like there's more that are in this family, and that some people are testing when they're testing for psilocybin, but we're just starting to get to know them. This one is actually pretty similar to the one, an active compound in the toad licking, that the toads secrete, bufadiene. This one is very similar. These two are suspected to not actually be bioactive, but to be precursors, and we just don't understand the pathway yet. And so I put these up and I talk about the other molecules to say it's a vast oversimplification to talk about psilocybin percentage, and then that's the potency of these mushrooms, or that psilocybin is the key, the psilocin is the key one, because they act in conjunction. They are, and like, so imagine this exists in the mushrooms. The bioactive form psilocin is also present. So imagine the difference between a mushroom that had a lot of psilocybin and a lot of psilocin versus one that had a lot of psilocybin and no psilocin. Your onset alone would be different, right? You're consuming something that has a bioactive compound in significant percentage, as opposed to the one that you only are consuming the precursor and have to wait, right? So they interact, there's synergistic effects between them, and it makes it very, very complicated. But there's a good analogy for it that I really like. Paul Stamets has given this analogy on 2 or 3 podcasts that I know of, and probably dozens of times. And he relates that someone told him this, that someone who had taken both lab purified psilocin and consumed mushroom, fruit entire, made the analogy that the lab version is like a box, and that you go from baseline to maximum elevation and almost immediately, and then you spend all your time at that, and then you're back down to baseline. Whereas taking a whole mushroom is like wandering through a hill, right? You kind of like, and I think this is a good way to illustrate how those other compounds interact with each other. And when you separate out this kind of purified or even synthetic form, it's a different experience altogether. So I like that as a representation, a way to think about the way these all synergistically interact. But we are just going to focus on psilocybin when we talk about the concentration and the potency, because it's a vast oversimplification, but it's necessary. I mean, even people who this is their life is researching these things, it's complicated when you get all of them in the variable percentages. So we're going to just talk about psilocybin, even though we understand that there's a family of compounds. The next thing that makes it complicated, besides the fact that there are multiple compounds and the ratios can vary, is concentration of psilocybin varies massively. So this is one of the key things that does everyone know that? Do people familiar with this? Like, I find that most people don't, and it's the number one thing that I want to express to people. I think that they should understand that when someone says you should take 1 gram of these mushrooms, or you should take 2 grams of these mushrooms, or 1/2 gram of these mushrooms, imagine if I said that about chili peppers, right? What if I said this recipe needs 1 cup of chili peppers, but I didn't tell you if it was shishitas or habaneros? Those are very different experiences of eating those, right? So imagine that every time you're getting some mushrooms, some psilocybin mushrooms, it's like a mixed bag of peppers. And sometimes it is going to be mostly habaneros, and sometimes it's going to be mostly shishitas, but you stick your hand in there and you're not quite sure. So we're going to talk more about this variance in concentration, but that's another reason it's pretty complicated to kind of understand the potency of psilocybin mushrooms, because there's a family of compounds interacting with each other. They come in variable ratios, and the percent of the main, the percent of the main biocompound psilocybin and psilocin is the great variance between the percentages of them. Okay, so how do we measure psilocybin? When I say like we have a percentage, or we know, how do we do it? The main way is something called high performance liquid chromatography, which we're going to look at in the next slide. Mass spectroscopy is also a technique that's used in conjunction with HPLC, and like a little bit, like if you were a mass spec lab and you wanted to look at these mushrooms, that would be your tool, but it's not, you don't really need to understand that to understand the scientific literature on these mushrooms. And then the other thing is a colorimetric, which is like a pH strip, right? Like, so you have a pH meter that will tell you the 2 decimal points, and then you have that strip where you're like, I think it's kind of orangish yellow, right? And it tells you the pH. There are colorimetric assays for these, but they are very kind of qualitative and kind of estimated, and they are also, pH strips are everywhere. the colorimetric assays for psilocybin are, as you can imagine, because it's a controlled substance in some parts of the world, there's not very many. And getting a decent one, there's subject to temperature fluctuations when shipping and stored and this and that. In general, they're just a very rough guess unless you're in a research lab and developing your own colorimetric assay. If you're trying to buy them, they're hard to find and not very accurate. But let's talk about HPLC, because this, in addition to the clinical research that told us about how humans interact with these mushrooms, and scientific advancement we made was being able to use HPLC. It's a technique, not like new knowledge, but using HPLC for these mushrooms has helped a ton, both in the research lab and in the gray market, like citizen science. Okay, so this is HPLC, the general idea. There's a column. The beads are obviously not like that. They're very, very tiny. And they have some property. In this case, very commonly, they have a charge on them, right? So now you put this like mixed bag of stuff in there. And depending on the polarity or the charge, like the properties of the mixed bag, they stick to the column with different degrees. They move more slowly. So at first, nothing's coming out. Then the blue is coming out. Later, the white, the red. Does that make sense? So this is the way, instead of looking at like a bucket of pond water, you can look at like water with one bacteria in it, right? You know, like it's clear, it separates it out and lets you study it and understand. So you can separate all those molecules that are so similar. Like one of them is, you know, some of them are just a few bonds different. You can separate them out clearly and look at exactly how much you have. Like, if you were trying to know the number of squirrels and types of squirrels in a 10 acres of forest versus the number, you took all those squirrels from 10 acres of forest and put them in the room here with nothing else, right? It's like, it's a lot easier to understand the compound when you can separate it out this way. And the proliferation of this technique across citizen science and things has helped a lot. Here's just one more illustration, because it's easier to see. I recognize the one on the left is kind of like not very high contrast, but this is the kind of general idea. It helps you separate things out. So let's talk about some ranges, right? So using this HPLC, we look at these mushrooms, like what kind of range of psilocybin concentrations do they have? This is an example of some wild mushrooms. It's a characterization paper. Alan Rockefeller is one of the authors. And so they went out and they collected samples of all these mushrooms, and then they characterized the percentages of psilocybin and several other tryptamines. And it's just the early stage of getting to know what's out in the wild. So I know this table is absolutely unreadable, so I pulled some things out, but you can see on in the small print table, that's from the paper, and it lists all the samples that they have by genus and species, and then gives the ranges for various tryptamines. I just pulled a couple things out to show these are so within one species, right? Let's look at the bottom one, the one that says between 1.55 and 15.54, 20 mushrooms, and that's the range. 10x difference between which of those 20 mushrooms you picked up and ingested. Right? That's pretty significant. That's going back to that peppers analogy. That's like between a habanero and it's not just. So then you might say, well, okay, these could be weird exotic mushrooms, and who knows where they're from, and like that's not the way it is. And if I went to like get mushrooms from a dispensary in Oregon, right? That's just wild mushrooms. But okay, so let's look at mushrooms in the market. And for mushrooms in the market, we're going to talk about this thing called a psilocybin cup. People who are familiar with cannabis will be aware that there's things like a cannabis cup. It's the same idea. So this is a great source of data because, first of all, they're running really pretty good analysis. They got good equipment and they know what they're doing, and they're running pretty intensive analysis against all the samples. This isn't like the state fair where you just taste the pie and people vote, right? Like they are measuring and analyzing these things. And then another thing is this is a gray market thing, right? It's in Oakland, but it's not federally legal and this and that. So it was underground at first, and it was not the case that you could just sign up and say, I'd like to get my thing tested and throw my ring in the hat. It was known and respected mycologists within certain communities were invited in, right? So I mentioned that to say that this data comes from a subset that is experienced and respected cultivators. It's not just a mixed bag of like somebody who just learned and somebody who's been doing it for a long time. So these are, I'm trying to justify that these are representative samples. Okay, So here's the kind of thing I used an early one because this is from 2021. And I use that because the way they look now is like this. They're a lot more complicated. This is the same organization, same contest, but over the time, they're getting more sophisticated in their analysis. So for us to look at that, it seems like a headache. It's a lot of going on, right? So let's look at this one. This one is just testing for psilocybin and psilocin. And it has the name of the people who made it, a name that they might have for like this variant, and then the strain. And it has percentages over here. It's still pretty small and hard to read that from wherever you're sitting. So I pulled some things out. And I know the name penis envy is silly and it might make giggles, but it is a very popular one. And the reason I picked these subsets out is golden teacher and penis envy are two very popular and well-known types, right? If you're trying to find some psilocybin mushrooms and someone says, I can help you, or you can go to this dispensary, here are all the strains. If you know any names, though, you probably know they're very popular. They also have a reputation. Penis envy is considered to be extremely potent, and golden teacher is considered to be very mellow. But you can see right here that there's a golden teacher that's stronger than one of the penis envies, right? You can, and then even here we have like the same people growing the same strain, and we're still having that kind of difference, right? So then again, I'm trying to drive home that there is a vast range of potencies, and you don't really know, unless you have the testing, what you're in for. And then I also like this one because this thing called tidal wave was this fall 2021 was noted for that. Previously, the understanding was that the maximum that could be was 2. And then there's some things that are like 1.8, you could get 1.8 and maybe you could get 2 and this and that. And then this tidal wave just blew past that. And yeah, it was a big deal. And then this is just again for like the whole range between that. Okay, let's see. So this makes a difference when you go to consume it, right? So let's look at this. If you have a penis envy that is 0.6% by weight versus one that is 1.7, that's going to make a significant difference in how much you're ingesting, right? And so this is just kind of a graphical representation trying to drive home how much variance there can be in this. And then there's one more factor though, and that's body mass. So if someone who is, let's say 120 pounds and someone who is 180 pounds, and they're both going to eat the same amount of any of these mushrooms, they're going to have radically different experiences. Okay. Now I'm going to introduce this concept of milligrams per kilogram, because that's how we really want to talk about concentration. And when you look at the science paper, that's what they'll tell you. We gave 3 doses of low, medium, and high, and they were 0.1 milligrams per kilogram. They're adjusting for body mass. And again, so we'll talk more about the range that they use in the clinical studies, but I'll tell you it's commonly 0.1 to 0.4. So imagine I'm trying to reproduce. I read the paper, I've seen a fantastic fungi. I want to have this connection with nature, this experience, right? And I'm trying to replicate what I read in the science papers. Well, depending on which of these I have, this is like hardly going to be detectable for this person, and this is going to be very high, right? So we'll leave milligrams per kilogram for now until after we talk more about the science papers, but this is, I think, an important part of concentration is not just a lot of guides and a lot of like popular blog posts and things will just say, oh, you should have a 0.5 gram for your first experience with this mushroom or something. And I think this shows that it's a lot more complicated than just the weight of it. Okay, so let's talk about some research in clinical things. Didn't I have a bottle of water? Okay. Oh, sorry. Okay, so the first thing I want to talk about is neuroplasticity. Oh, thank you so much, Sean. Is neuroplasticity. So that which includes the concept of neurogenesis. Isn't that pretty exciting, just that word, growing brain cells, right? You know, like that was not a thing when I was like up into high school science. It's a fairly relatively new thing. We don't grow new fingers when we cut them off, right? You know, like growing new brain cells was not the understanding of neurology in the 80s. Or even maybe until the 21st century. But so that's pretty exciting. And I just want to say a few things about when they study neuroplasticity, they study a lot of things. They study the biochemical pathways that can produce it. And then they study it specifically kind of by, can everyone read this? Yeah. So like you can have different, you have neurons forming, dendrites forming, and synapses, like new connections forming. These kind of mushrooms and the compounds in these mushrooms promote all of these things. Imagine like the potential for like Alzheimer drugs or brain damage from other, things. That's, I think it's very exciting. Like I said, just the idea of neurogenesis makes me very excited. I'm not going to go into these too much because there's not really one standout, and they tend to be either they're preclinical with animal models a lot. But this one is a pretty big one that kind of nailed things down. This really cemented our understanding of how these compounds in these mushrooms promote neurogenesis. And they have a great graphic about it, which is, so yeah, the graphic speaks for itself. And the psychoplastogen is a word for things that can cause neuroplasticity, and it's specifically a psychedelic. So in this study, they not only looked at psilocybin, they also looked at LSD and DMT. But this is the concept of new things can be growing. Another paper I'll show very briefly because it's early for one. It's 2006, and this one is kind of neat. They have two ways that they're measuring this. One is it's an animal model, so they put a fear response into an animal, right? Like every time a bell rings, you get an electric shock, and then when the bell rings, you get scared after a while, even without the shock, right? So they condition that into the animal. And then they give them psilocybin, and then they see who can get rid of the fear, right? And most of the animals without psilocybin, which is saline, they'll have that fear response. If they never ever see that electric shock ever again, they will have the fear of that bell for the rest of their lives, right? And I think we can all kind of relate to that, right? Like maybe you got like a little bit, something a little challenging, and it's still kind of tough to get around, or a habit that we want to break. And why is it so hard to break that, right? But then when they gave the animals psilocybin, they lost the fear response. And then the second part so they look at that behavior in the animal model, and then they cut open the brains, and they can actually see growth with like fluorescent labeling, and they can see that there actually is new growth there. Okay, so let's move on to clinical. That's kind of this background that neurogenesis is possible, and evidently psilocin and many other psychoactive compounds are promoted. So that's pretty exciting news and a novel take. So here's some things. This is definitely not the definitive list of second wave psychedelic research. That would be an argument, and it would be 12 to 20 things long, but let's look at a few of them. So the first one I want to look at, again, because it's early and it's pretty impressive. So this is looking at obsessive compulsive disorder, and the introduction of this paper makes it clear that it can be debilitating for people. It's a very serious condition. It's not an inconvenience. It is, disrupts your life and things. And it is notoriously hard to treat with combinations of drugs and talk therapy and other psychotherapy. It doesn't have very good results in general. So this person at the University of Arizona, the dosages they were using were there. So this is significant. This is a psychedelic trip. This is not microdosing, right? This is a significant amount. And it was astounding. First of all, every patient had a decrease. That's very rare in a clinical study that you see 100% of the sample set responds positively to it. There's, even if you had 50% of the people got kind of better and some of them didn't have anything at all, it would still be exciting, right? Everybody got better. And they had, some people had a total absence of OCD symptoms. There's a test for this. It's statistics and it's testing and self-reporting. So it has some, issues there. It's not, but it's not, how do you feel today, right? It is a very complicated, thoroughly studied, well-designed, rigorous test called this Yale Brown Obsessed. And imagine like the surprise of these researchers, like I had to say, if you had like some people who are 100% recovered over the course of your study, breaking these very, very hard to break ingrained patterns. I know, I find it very exciting. Here's one. Okay, we hear a lot about this, right? How about people who are facing into life or or illness issues, and they have related anxiety and depression, right? So here's one in right, this is UCL, this is the UCLA one. And so they did single dose. Again, it's not microdosing, it's a significant dose, where it's 0.2. But they had these increases that were really impressive, I gotta say. And both is the thing. Like within the patients, they had both these improvement in both areas. So it's, not quite the same as like as impressive as the total absence of OCD symptoms. It's still notable increase. And the other thing is they last. It's not like you take this, if you think about antidepressants or some kind of drug like that, tranquilizers, the sedatives, you take them and they make you feel a certain way, and then they're out of your system, and that's it. That's not the case when you have psilocybin mushrooms. People are maintaining those benefits 6 months out, probably longer, but most of the studies only do a 3 and 6 month revisit. Oh, tobacco use. Talk about, this is a serious drug. I mean, I think they talk about this being very, very hard to put nicotine. You can see like the billions of dollars people spend on it. And we normalize cigarette smoking, but it's a very addictive chemical. So anything that can beat that is, got some power and got some interesting things you want to know about. So they did 2 to 3 dosages, and it's on the higher end compared to some of these other studies. But again, what did we learn about like potency? These are pure psilocybin injections, but so aiming at this is just difficult because of the variance in potency. But they're on the higher end, and 80% of people aren't smoking. No other program that had ever been seen, the best that they could do with, in any other program was about 35%, and that was considered serious. Like, great news. And think about people who smoke cigarettes, who consume tobacco. They quit and they start, and they quit and they start, and they quit, and you know what I mean? So having 80% cessation is amazing. And this really, to a lot of people, stood up and took notice about this too. And it's one of the Johns Hopkins studies that got a lot of popular press. And this is another end of life one. And I think this one is UCLA. So what do we see here? Okay, this one I love. This one I love because this is showing that the low doses do it too. This is another one that got a lot of popular press, because previously what we were seeing was, we're going to have this experience. It's kind of a high dose. It's a big deal. They often have counseling leading up before it. They have a guide with them there. It's considered a very big deal to give you this high dose. And I think for lay people, some people might be more hesitant about having a full-on psychedelic experience versus taking a small amount that may or may not be detectable, right? Like low doses, that's exciting if you can get kind of the same effects as low doses. And it turns out that you can. The red line is high doses, so there's a scale thing, but you're still getting a single dose. This is a single dose of a small amount of this compound from these mushrooms. And you're having over time, these are 6 months if you can't, I know there's a lot of frames in here. This is over time, you're seeing this kind of improvement from one dose of a small amount of this mushroom. It's a purified and or synthetic version, but we know it's a mushroom. This is, okay, how about this? Like, this one is about, it's called like persistent depression something, or like, it's severe depression that is not considered untreatable. Right? So if you imagine all the people suffering from depression, some of them are going to do some course of therapy for 6 to 18 months and be better forever. Others, it's like, it's a chronic problem throughout their life. There's an intensity spectrum too, of like, this is an intense, severe depression that has been resistant to all other kinds of treatments, and the people are just, right? So this seems like a great thing. If you think you have a new superpower in the form of psilocybin mushrooms, let's try it on something very, very tough, right? So again, just 2 doses, right? It's not something you take every day or anything. And it was astounding. Everybody had an improvement. This study would have been amazing if like half the people had a response. We're talking about very severe cases, right? And everybody had an improvement. 25% of them were in remission 5 weeks later after a single dose of this compound from this mushroom. Okay, well, that's my, do people have any questions about the research thing?
Yeah, how does it work? I mean, it's doing all these magical, cures with psychological cures
Yeah.
in the body. Do they have any clue as to,
Mechanism.
yeah,
I'm so glad you asked that. So I didn't make that clear enough. Then, yeah, I definitely did not. The neuroplasticity is why these behavior changes can happen.
okay, so it's
It's growing new, like
regrowing.
growing new neurons, dendrites, and synapses in your brain.
Awesome.
So that's how you can change your behavior so much easier. Yeah, I think I just was like neuroplasticity, all these clinical results. Like, that's the link between them. That's why. And so by growing these new connections, there are literal physical ruts in your brain from certain behaviors. Like your thoughts and your processing of the stimuli you encounter makes physical changes in your brain, in the squishy part in your skull. And there literally are physical ruts sometimes. But if you can change the shape of that, then now you can change your behavior.
So it's kind of like the mycelium in the earth.
Mycelium and synapses seem really similar, don't they? Yeah.
Right.
Thank you for asking that. I'm glad we clarified that. So the last bit is, oh, sorry.
Thank you. Can you talk a little bit about what accompanied the dosing? Like, it wasn't only taking a dose.
Sometimes it kind of is, but yeah, that's a great point. A lot of these studies, it's not really like, it's definitely like you walk in and sign up and you take your psilocybin in a cup and you go later bye and then you're all better. Like they're combined with things like cognitive behavioral therapy. So in some of these studies, like the tobacco one, sorry, that's an alcohol one. There's an alcohol one in which there's 4 weeks of intensive therapy before they ever get to the psilocybin. And when you chart their results on these kind of like indexes of depression and anxiety, you see a minor drop in the beginning, right? And then you see a substantial drop with the psilocybin, but there's no question that the therapy was very important to it. And then there are some that are a little more straightforward. They're monitoring your vital signs and they're asking you questions to index about how intense your hallucinations or your sense of time or those things are, but they're not quite doing that really close connected, intense long-term therapy. It could just be a couple of sessions to prep you for your session and then a couple experiences or a couple things after. So it can be lightweight, but there's always something. There is always something. Yeah.
Well, I read a study recently that had to do with neuroplasticity in older folks.
Mhm.
And this doctor was maintaining that we're not supposed to have as plastic brains as we once did when we were younger. And that there needs to be ingrained things in our brains, especially for older folks, so we don't get confused. I personally think that's a lot of baloney because I actively know people who use the substance on a regular basis and they're just fine. But do you know anything about that, the detrimental effects?
No, I'm
on older people.
I'm not familiar with that. And as a concept, like that we need this ingrained pattern. In my understanding, none of these compounds affect long-term memory or retention.
Yeah.
So it's kind of more like habits and behavior. I can't see that you would be like forgetting facts that you need.
I'll find it and show it to you.
Okay, yeah. And I mean, I guess it's conceivable. So one of the things during these experiences is synesthesia. I'm close enough, right? Do people know what this is? Thank you, synesthesia. Do people know what this is? It's crossing of when people say they smell colors or they taste sounds. So that is something that happens during your experience, right? And I mean, I guess I could conceive that boundaries getting fuzzier in your brain could have some downsides. And it also depends on the person, I guess, right? Some people are about structure and routine, and some people are about a little more fluid. But I'm sorry, I don't have a very satisfying answer to that, Tom, but I'd love to look at the article afterward. All right, applications to everyday life. So how do you take these things and put them into your own life? the first thing I want to go back to is, oh, sorry. First one is, I want to go back to the milligrams per kilograms and looking at dosages. But first, I think a big question for a lot of people is, where do you get this medicine? Right? So you're interested, you've been reading about it, and you don't know anybody who does it. There is in Oakland something called the Church of the Zide, Z-I-D-E. It's a very interesting organization. As you can see, they have memberships, but it's very easy to get a membership. That's a little bit of like legal protection for them, and having members that they're not just like a store selling to the public, but they have cannabis also, but they are a mushroom dispensary. And they're in Oakland, and it's pretty neat. They call themselves the Church of the Entheogenic, what is it? Plants. The Church of Entheogenic Plants. And they really get into it. Like there is a church in the dispensary with pews and everything, and a little thing that you stand up in the front. And when Dave, the person who founds it and kind of leads it, presents in conferences and things, he comes in like bishop's attire. I mean, like he has like a hat, and he has like this thing, and he has these really fancy vestments, I think is the word, right? And because he's constantly trying to make the case of, how can you tell me what you can't regulate religion in this country or something. And they have also done a lot of work for, in general, just kind of around decriminalization, and they put themselves out there, right? I mean, if this is your organization, you have headaches all the time, right? You're like walking into headaches. So thanks to them for doing it. And it is a safe place to go. It's not that everyone who works there is going to be able to tell you about every mushroom in their catalog, but I will say that they vet their cultivators, and like you can trust what you get from them, right? Like they might not, if you go directly to a cultivator, you can learn a lot about the mushroom. And so at the dispensary, maybe not as much information, but definitely a safe and secure place. Then of course, Oregon. Like you can just drive to all of Oregon, you can get mushrooms in a dispensary. And I think, I don't know, it feels like there'd be some travel to that. It's not that far from here to Oregon, right? And then people go to Canada to get cheap pharmaceuticals. So, and then this is my number one thing, talk to your friends and acquaintances. Not just because you might have a good connection to get some of this medicine if you're interested, but also because we need to normalize it. And we need to there's a lot, everybody who's here came to hear a talk about these mushrooms, right? But there's lots of people who maybe read a newspaper article or watched Fantastic Fungi or read or watched How to Change Your Mind, and they have so much stigma, right? They think of like drugs are bad, and this is this is something bad and I shouldn't do it. So I think we need to kind of normalize it and talk about it more. And I've heard a friend of mine has a presentation on microdosing with his family and how much healing there came out of that, right? So he's in his 20s, and he convinced like an aunt or a parent, and then slowly more people, and then people are suddenly talking about their feelings and talking about their goals for microdosing. And I'm getting chills just thinking about it, that it's just a beautiful thing, right? But again, it kind of needs to be destigmatized still. So if you make a small talk with someone, say, I read this newspaper article, I saw this talk at the Mushroom Club, and have you ever tried that? Have you ever thought about it? And yeah.
I just wanted to ask about microdosing. Do you have any psychedelic kind of experience when you're microdosing?
No.
No.
And we're going to talk a little bit about scale, and so we're going to talk about scale in just a second. But yeah, ask your friends and acquaintances, normalize it, share information about where to get good medicine. And okay, so scale, very next slide. The title of this used to say scale, and then I kept writing with 100 caveats, or with many, I don't like the mass scales, because again, 1 gram of one kind of mushroom is very different than 1 gram of another. But we sort of need a framework, right? We sort of need something to get started on. So this, I don't think anyone would really argue with this. They might draw their lines a little differently, but nobody would tell me that this is, there's a thing called a nanodose, which is absolutely not detectable or detectable or perceptible. It does seem to have the neurogenesis effects. So for people who have a little skittish about the idea of psychedelics, whether it's from stigma or just they're not interested in a psychedelic experience, a nanodose could be the way to go. You're not, even on an empty stomach, even on all the conditions lining up, it's not going to be detectable. This is a pretty good range for microdoses, I would say. Again, like I've heard people compare microdoses of saying, oh, that's 100 milligram microdose of this mushroom. That's probably about equivalent of a 200 milligram microdose of this mushroom. And the people agree, you know, like, but that's at 200, you are risking detecting, it's absolutely not psychedelic, but you might feel something that's like a little extra energy. You might feel like a little things, a mood booster of a bit. People I know have talked about, they take their microdose and 20-30 minutes later, they feel like they just had a coffee and, you know, or something like that. They feel a little, but that's not psychedelic effects. There's no visual effects or things like that. It's just a mood enhancement. 500 milligrams, again, you know, roughly, I really think this is a really neat dose. This is somewhere where you're starting to have a psychedelic, just a little bit of a psychedelic experience, but it's absolutely not the full on experience that. And so this is a way to stick your foot in, like, you know, kind of wade into the water and see. Did I just unplug something?
Yep, that's all right.
Does it have your plug?
No, it's good.
So this is kind of an intermediate here. And some people, they take a couple, 2-3 of their microdoses or something. A lot of chocolates and other things come in this dosage of about 500 milligrams, about half a gram. And it will be a mushroom experience. It will not just be a general mood elevator or slightly more energy, but it's very mild. So that's a neat range for people to play in if they're a little nervous about the idea of psychedelics, or if they've been microdosing, but they've never taken a larger dose. Once you get at 1 and 2 grams, this is when you start having a psychedelic experience. 3 to 5 is very high in my mind. There are people who do extremely large dosages. In fact, Church of the Zayador, that is kind of their philosophy, that the goal is to take very high dosages. And I don't know. I don't find it interesting. No one's studying it in the lab really, but there is a percentage of like a subculture within the psilocybin mushroom culture of people who want to take so much that there are physical limitations, you know, like that they have to empty their body because otherwise they would get emptied during the, you know, like if you take too much of psilocybin mushrooms, your body will evacuate your bowels. So there are people out there who put themselves in it, like they don't eat for a while and then get ready and they have some people there to make sure they're going to be okay. And then they take an extreme amount of mushrooms, but so I feel like anything over 5 is getting into like kind of, it's like, I bet everyone in here kind of likes hiking, but how many of us really want to do like the whole like Appalachian Trail for months, right? You know, like that's just a different category. And again, there's not really anything in the lab on that. So lots of things have inflection points. If they're good for you, they're good for you, and then they're not, or something, you know. We're going to Tom's point about maybe too much plasticity isn't the best or something. Yeah, so we don't have any really science.
Your scale there is talking about cubensis?
Pardon?
Your scale there is talking about cubensis?
It is not. Thank you for clarifying. So do you remember in the beginning when I said there's all of these different kinds of like genus and species of psilocybin? That's why the scale is kind of useless. Because even if it was cubensis, there's a huge range within cubensis, but no, it's supposed to incorporate everything from like every kind. It's not. It is considered a scale for mushrooms, which I find just, it's hard to understand. Just like if you told me like this is the scale of like of chili peppers, and then you mixed a whole bunch of different kinds of chili peppers together, right? Like having 1 chili pepper is great. Having 5 is a lot of chili peppers. Well, dude, that's not true about chili peppers, right? Anyway, we need some sort of framework. And another note I want to make about this scale, if you're looking at products, so I said, where could we get medicine? And I said, you know, there's a dispensary in Oakland and there's all of Oregon, and you can talk to your friends. There are online things, which I strongly advise against, just because it's anything off random sites on the internet doesn't seem like a good idea, right? Whether you're talking about chocolate chip cookies or psilocybin mushrooms. And one of the things about those kind of products that are just like available is they're often in stacks, which means the person who has made them or the company that has made them has decided that the best possible benefit is psilocybin mushrooms combined with lion's mane, combined with this, combined with that. I think it's it complicates your ability to detect your response to the substance. There's interactions between those things, right? So if you're trying to get to know mushrooms and learn if they will work for you, I recommend just microdoses or being just psilocybin mushrooms, just ground mushrooms, and not any of those other additions. Okay, so now we're back to this like scale. We learned what the scale they were using in the research papers, and let's talk about how it translates out. So this is the same one I used before, just showing that like depending on the concentration in your body mass, you could end up with a very different milligram per kilogram. Let's go back to this like example we had from kind of market available mushrooms. And just do, because decimal points are tricky, and I like to see it kind of written out. So the unit that those folks use is percent by weight. But if you want to translate that into milligrams per kilograms, like if it's zero, let's just look at the bottom golden teacher. If it's 0.4% of the weight, then that's like it would translate to, I just realized my poker wasn't showing. If it's 0.4%, then this is how many grams of it are in a gram of mushrooms, and this is how many milligrams. Okay. Maybe that was superfluous. I just, like I said, I like to see things written out. Okay, so here's our list from the fall 2021 psilocybin cup again, and we've put in milligrams per kilogram for a 120-pound person and a 180-pound person. But that list is like not ordered, and it's maybe not as easy to compare 14.4 to 11 or something. So let's just do this. Okay. If you had, this is like that tidal wave, right? This is off the charts high. And this is very mild. In my experience, I would say that most of the things on the market are probably falling around here, or like 0.75 to 1.5 or something. So if you wanted to reproduce the study and you read the study and you said, okay, well, what they did is they took these tests, these kind of multiple choice tests to assess your depression, your mood, your anxiety, whatever, quality of life. And then I'm going to take the same amount of mushrooms that they took in that study, and then I'll take the test again or whatever. You would, so remember, this is our scale. Well, that's with the very, very tiny ones. Remember the one I showed when I said it's so exciting that just the micro can do it. So mostly it's 0.1 to 0.4. So you'd be eating, if the person told you, these are pretty strong mushrooms, right? I feel like these are pretty strong mushrooms, then you would be, and you weigh 120 pounds, right? You would eat a whole gram to get to like the middle range. Is that clear? Okay. Here's some.
Those are dried mushrooms as well. You're not talking about fresh made mushrooms.
Absolutely not. Freeze dried usually. Yeah. So very, very cracker dry, very, very dry, no moisture in it. And then, so this is an example of how you would kind of figure this out and as far as like, where are you getting these numbers? People can tell you that strains test in a range. They can tell you this cultivator with this strain, they tend to test in this range or something. They can give you things like that. And they also just like the human experience of, in your experience, like, is this mild, medium, or strong? And then you can start kind of figuring it out from there. So I also want to say a little bit about microdosing schedules. We didn't look at any studies that had microdosing schedules. And I don't think that Fadiman, people familiar with this is a person who's a health person who's very keen on microdosing and has a lot of material about it. But I feel like it's mostly blog posts and white papers. There might be clinical studies from Fadiman team. And then Stamets, so there's nothing special about Monday through Friday. It's just 5 days, right? You know, like the block could be anywhere. But the Stamets idea is you take it for a block of time and then you have a pretty time off. And some people would say that that's easier for you to detect the pattern, right? And when you do kind of a 1 day on, 2 day off, maybe, you know, the first day was just a really like atypical day for you or something, right? It's harder to see the pattern, I feel like maybe. But a lot of people start with one of these and then they just go by feel, you know, like, and there's a little bit of calculations about how much we're talking about ingesting, which isn't very much, as you can see. Okay, so here's just some like helpful practices. And the one thing I would say about that's not on the list, but I would add is, in general, get a lot and like homogenize it, or at least have a lot of the thing, multiple months. So I just did pattern recognition, that it seems intuitive that the Stamets schedule would be a little easier to do pattern recognition with. And that's the same reason that you want to get a lot. Because imagine you're trying to figure out what this does for you and the benefits, if you like it and this and that, and you're trying a different one every time, right? You go and you get like a week's worth of microdoses or something, and then you get another, like, this is a world in which we can go to a dispensary or something, but it's very hard because this week might be very different than next week. So my advice in general is to get like a month's supply, so you can see what that mushroom is like, instead of varying the mushroom in addition to all the other variables in your life. So that advice is get a lot, and then the homogenization thing is, if you are getting it from someone and they have a batch of microdose capsules, they will be relatively homogenized. They blended up a lot of mushrooms and made it into a powder and then put them in the capsules. So the capsules you get, it's not like just getting a dried mushroom and hoping it's not a spicy one, right? So the homogenization thing is nice, of getting like a bunch of all one batch from your person who's your provider, or buying the dried fruit yourself and then grinding it up and putting it in so like it's all kind of mixed together.
The problem with grinding things up that are dried is that if they're not vacuum sealed, the potency of the product will diminish very rapidly in the powdered state.
Excellent point. And in general, they diminish rapidly in like any kind of heat or light or things like that. But there are steps you can take to get around that, with like dark mylar bags and making caring for them. But that's another complication in the potency thing, besides the vast difference between like one single flush and all these other things, is then how people treat and store them, right? So like, how long ago did they harvest those, and did they grind it right up right away and make like 1 million microdose capsules they're going to give out for the next year? Or did they so yeah, storage and care of it does definitely affect potency, like Tom said.
I asked Alan Rockefeller if they would lose potency if they were in a plastic bag in a jar in the freezer. He said, talk to me in 10 years.
That's funny. Yeah. And the freeze drying helps with that too. Like, so that's another thing, is freeze dryers are now available for people with some disposable income. But it used to be that freeze dryers were like for small factories and they were $40,000, $60,000. And now you can get one for like $3,000. So that is great for food. I'm huge with freeze drying for food, but for anything, instead of like 2 days or 1 to 3 days, let's say, at a low heat at time, it's done, right? It's like it's frozen and it's dried and there's no water in there, and it's kind of like it's like a flash stop of nutrient loss, right? Like I eat a lot of freeze dried orange slices. You can eat the peel. It's really neat. It doesn't taste bad, and it's got all the nutrients. I'll bring some for the next Mushroom Club meeting. All right. What did you say? Oh wait, Pat, like I was gonna bring kombucha, but I'll also bring freeze-dried fruit.
One thing I was gonna mention too, with the microdosing schedule, I know the Fadiman schedule is more so to combat the like short-term tolerance buildup you can get from psilocybin. And then the other thing too is like people with stomach upset, depending on how you're taking it. I've had a lot of people say chocolates are usually better, as far as if you have a sensitive tongue.
Excellent point. So this is definitely something you should think about in applications to your everyday life. In the lab, they're getting this synthetic or purified form in a little pill. But when you eat dried fruit, and sometimes even when you just swallow ground up dried fruit, some people will have a stomach reaction to it. And so that's a consideration. Maybe you take it with food. Maybe like Stephen said, you mix in with chocolate is really popular. And there's a lot to the chocolate. Like Mexican people used to, like historic Aztecs, Mayans, they used to mix it with chocolate. But yes, it can cause an upset stomach, and yeah. So
What about lemon juice? I've heard of people doing it with the lemon juice chaser. Is there?
people know this lemon tech thing, yeah. So the idea of lemon tech is what some people say, but with lemon juice is you have the ground up fruit and then you squeeze the lemon over it and then you throw it back or, you ingest it from there. And that the onset is different and some would say the experience is slightly different. It is not understood very well and there's not much research. I suspect that it's about the acid. There is some research on like acid preservation and degradation in different pHs and things, but no one really knows. At the very least, it's the onset is different. Like it has maybe the acid environment has dephosphorylated or done some of the work that the enzymes in your body would have to do, has done it in advance, but the mechanism is not understood. In my mind, that is like a bit of like a novelty thing for people who take a lot of mushrooms, maybe. Like I wouldn't just say that someone would just always do the lemon thing. Do you know what I mean? It's like if you already like alcohol and sometimes have cocktails and things, then maybe you would experiment with absinthe lit on fire, right? But you're not going to drink absinthe lit on fire every day, right? Comes on a lot faster.
First of all, a lot of times what you want to do is grind the mushroom, put the lemon juice on it, let it sit for a while, strain out the physical body of the mushroom, and just drink the juice. And that's a much cleaner kind of uptake. You don't get there's hardly any body response. There's never a stomach response, and it comes on instead of waiting for an hour to digest the mushroom that comes on in 50 minutes.
So the onset is faster, and a lot of people like that. It's just like doing shots of alcohol, right? Like a lot of people, I like doing shots of alcohol now and then. Yeah, it's fun how it comes over really fast. I guess what I'm just saying, right? If you do a shot of alcohol, that's a lot different than like sipping a hard cider over the course of an hour, But so I only mean to say that I feel like it's sort of like maybe a more advanced technique. And if you're like,
it's probably not recommended for really small microdosing because you just lose.
oh, it'd be a hassle. It'd be a hassle with those tiny little powders. Yeah. But even if you're thinking, all right, I'm ready to do this kind of like larger, like dose experiment I read about in the science papers, I just think it's not necessarily your first step. Yeah.
I would personally highly recommend using the lemon tech because it's just a much better situation for you. Like I've done mushrooms many times.
Yeah. But I also think that it's better for you, not necessarily better for everyone.
Well, they just say everybody that I've ever talked to about this experience has a similar.
Okay, and I do not mean to be contrary, Tom, but that's still anecdotal. Like there's a lot of difference in biochemistry and how we respond to these things, and it could be there's so many other factors that we have going on. So like, on this list of things is pay attention to time of day and food intake and things like this. Like, but yes, Tom highly endorses lemon tech, and try it if you're
Okay, so I just want to talk a little bit more about the, like the effects on the digestive system. After I've done, probably a teaspoon, which is, I don't know how much that is, but like, it's a full-on trip. Anyway, then, I didn't feel like eating for a day after. I mean, my stomach was a little bit upset, and I wouldn't say like hugely, but I just had no appetite.
So there are appetite suppressant facts. So like while you're having it, like there is a bit of a stimulant response. Like if you've ever noticed that stimulants in general decrease appetite, right?
Oh.
So like that's why like diet pills are speed, right? And like sometimes things like caffeine buzzes and things can decrease appetite. The caffeine pills were often in diet pills too. So there's a little bit of that while you're eating it. But I think I want to separate out these two things that we're talking about. The digestive upset where you have a stomach ache and it's physically uncomfortable, and maybe there's even like digestive isn't going like it does every day or something like that. And then this lack of appetite. In my mind, those are two separate things. Do people have experienced this maybe, or?
Well, I would say like it wasn't like I had stomach pain, but kind of a light nausea,
Mhm.
and then just felt like food was,
Yeah.
I was very.
Let's talk about this a little bit more after. We're almost done for one. Yeah. We want to just run through these last things. Let's take that conversation. Oh yeah.
I was just wondering if you have any idea you're gonna look for information about interaction between psilocybin and various psychotropic medications?
I don't have an idea of where to look off the top of my head, but there is information out there and in general, this is the information that I think you will find can be summarized kind of like, there's some sort of interaction. We don't really understand it. Don't take it. Like it's just a kind of like a vague understanding and like an abundance of caution of like, don't mix. Yeah, that's it. And I don't think so you can find a little more detailed information on that, specifically on the types of, what was the thing you said? Psilocybin and
Psychotropic medications, like antidepressants.
SSRIs and things like that. Yeah.
Or
Wait,
something like Parkinson's, or, just wondering about this category of.
yeah. So, okay, finding it on some common things like SSRIs, it's the only type of not antidepressant I know of. Sorry, I'm using it again, but like finding information on that would be kind of straightforward. Finding it on less common medications, I don't think, because it's not commonly researched, right? Like if it is a legal substance, then there'd be a ton of research on how it interacts, right? But because we're still in the beginning of the second wave and I don't think there's funding for doing research on extensively. So I don't think you'll find less common drugs, but you will find larger drug categories, right? They'll say this category of kind of drug interacts with this type of tryptamine in a certain way. But in general, it's not going to be satisfying in a lot of detail enough to make you feel like I'm comfortable mixing these two things because I now understand it. It's going to be like some vague things and then a warning not to mix them.
If you were gonna put yourself in a position to be getting the information as it comes down, where would you look? Is there any particular research organization or any particular Johns Hopkins or Mayo Clinic?
Well, I would look at the papers rather than the institution. So yeah, you could subscribe to Johns Hopkins, and then when they have, they've got mailing lists about their science or their university, and when somebody publishes an important paper at Johns Hopkins, the mailing list will send out, and I get the Berkeley one because I went to Berkeley, and they're always sending out announcements about this cool paper came out in history or this, yes, Billy?
I went down the rabbit hole on this very subject. I was trying to help my mother out. She was on antidepressants and some other stuff for pain. And it's true, it was so hard to come up with information. There's not studies being done for all kinds of different reasons. I seem to find the most help from just people's personal experiences that I'm sharing. And I'm seeing a lot of those kind of discussions on shroomery.org. You can search discussion dialogues within that site. And basically, you're just tapping into a lot of individuals' personal experiences. It was definitely a concern, obviously, for trying to help my mother out. And it kind of almost feels like, well, we're not seeing a lot of repercussions or negative talk about somebody taking microdose or mushrooms while they're on these other medications that's having ill effects or things that counteract whatever medications that they're on in a negative way. So I kind of started to lean towards, well, probably not that big of an issue, but let's start off slow here and see what happens. And every body is different, and our chemistry is that's the whole thing with this body weight, dosage, all that stuff. It doesn't matter. I'm 200 pounds, and if I take a microdose, I've got a cousin that's barely 100 pounds, and she's like, oh, I don't really feel anything. And I'm like, an hour into it, I'm definitely feeling something. So even the body weight thing doesn't even really match up. I think it's in experimenting with microdosing in general, I think it's just really important to, one, have the product be consistent. So the exact amount and the exact strain and preferably coming from the same fruiting body mass. And then also just like paying a lot of attention to your body along the way. And to me, that also means like maybe weeding out maybe some bad habits like alcohol and other things that you don't really need to convolute it all out with, to be able to really understand like how your body is reacting, and also keeping like a specific regimen. Like if you're taking one microdose a week, you're kind of not really microdosing. Like you took a microdose, but you're not really microdosing. You want to have some kind of consistency along the way and be able to map how that correlates with your own body, because everybody is different. Size, what you ate that day, how much liquids you took in, what you're doing. Like, are you laying in your bed with your eyes closed in the dark, or are you out walking and expelling energy going up a hill? Like all these things like play into what the effects are going to be and what you notice. And some of us, as far as like how we take them and getting upset stomachs, I don't have any experience with losing appetite for a long time, but it's like humans kind of have this tendency to like always want to be hungry. And sometimes a little bit of discomfort is like part of the healing. And sometimes it's also good to embrace that discomfort and move through it, get past it, and learn from it too. So it's not always about like taking it with something yummy like chocolates or doing the lemon tech, or a lot of times it's just like eating a whole fruiting body and chewing it, then chewing it up for another 5 minutes after that, really pulverizing it. And so you don't have these bigger particles kind of sitting in your intestines and your stomach, and that's actually causing the discomfort. A lot of powder, like when you powder it, or you make cheese and you sift out the mass like Tom was talking about, that can be a lot gentler on your stomach and have. But I just want to say that a little bit of pain and discomfort isn't necessarily a bad thing, especially when it's like you're trying to get specific results that are going to benefit you in the long term. So anyway, just going back to that, like there's not a whole lot of information on, because these different pharmaceuticals, this drug with this mushroom, it's like, well, is this drug making company funding that research? And then there's so many different drugs. You have to do one study for mushrooms with that one specific drugs, and we're just not anywhere close to that. So a lot of this, the intel is coming from people's firsthand experiences that are sharing that information. And unfortunately, some of these more underground sort of websites like trumery.org, I mean, anybody can go there, it's there. But all this, a lot of this information is still vague. And like I said, everybody is different, so you just really need to pay attention to your own body. Start off small, start off easy, slide slowly. So that's my input on that question.
Excellent answer, really. Thank you. Wilder?
Sure, I totally agree. But just another tip for how to use the internet. Once you've done your due diligence on researching a drug interaction, you can set a Google alert, name of the drug, psilocybin research, and then it'll send anything that pops up on the web to your email inbox that has those keywords in it. So you might want to set a few with different words. That might be helpful.
That's a great thing. And my answer to it was going to be like where to go to look would be very similar, which is look for the research paper. So go to ResearchGate, go to Google Scholar if you're a Google user, like, and then the same kind of thing as a Google alert, and just go and see what came out new. But I still want to like reemphasize Billy's point that there's two things. Like we can read all the articles that we get and have nice tricks for finding them, but the firsthand experience in finding either other human beings like in person that you talk to, or on things like shrimmery.org. Yeah.
Okay, so, it seems like.
Oh, I'm so
everything-
sorry. I was mentioning the person behind you. Maybe start with her because we've had a few
Oh.
questions. I'm sorry.
Oh,
Can you,
okay.
can we just start with her? Yeah.
Yeah, I'm really enjoying this discussion. It's fascinating. Another resource might be PubMed, which is.
Another way to look for research papers? Yep.
Yeah, that's like almost like the resource, the biggest resource almost for medical research papers that have already been published. And you can put keywords and stuff. But my question has to do with, we were talking about like these papers are sort of sourcing the experience that these people are having, how the drug has helped them in conjunction with some kind of therapy. And I have some therapist friends, and it's a little bit like, they're talking about it more and more and more, and it's getting very exciting. But I'm curious about if there's any places or resources that you recommend that you can do these in the safety of, and where you can actually have a therapy experience.
Right.
You know,
All right. Does everybody get the question? So like, where can you get, there's a couple different levels. There's people who do psychotherapy with you, and then they also incorporate psilocybin. There are people who are sitters, which means they will be with you during your experience, but they're not really guiding you, and they're guides, so they're all different types of people that can assist you and support you and get you through these kinds of experiences with like a lot of love and attention. And I think that in this community, there are people who offer that kind of guiding and sitting kind of experience. We could find that a lot of that locally. I'm not familiar with anyone in Mendocino County that is like a licensed psychotherapist who does a course of therapy with psilocybin, but it does exist in the Bay Area.
but yeah, we'll talk after.
And then, Arcata, I forgot about Arcata. Arcata is also like Oakland, one of those places where it's decriminalized. And Arcata does have, they have like a group therapy program where they do integration circles and things like that, but not quite the one-on-one like therapy. So you might have to travel for that. And if you have therapist friends, then maybe you're the one who would know when we first start having some Mendocino County therapists doing it, eh? Are any of your friends thinking about offering it?
Not yet, no.
Okay.
I'm kind of slowly turning,
Yeah.
slowly turning the wheel towards the possibility of thinking that this actually works.
Well, I think that, yeah, like I said, in this local area, I know that there are guides and sitters. And then as far as like licensed psychotherapists, there's some nearby. One thing to watch out for is there's a lot of programs out there right now that offer to train you as a psychedelic counselor for a lot of thousands of dollars. And it's unclear about like necessarily like the value of some of these programs. And it's just a hot topic right now. And I think if you did a very service Google search or like internet search kind of thing, you would come up with a lot of things that were very expensive and maybe of limited value. So it's just, yeah. Yes, Sue?
Just a few things I'd like to mention is, first off, and related to your question with medications earlier, I've had experiences prior to taking medication and after. I have ADD, so I take ADD meds. And I do have to say that my experience with mushrooms after the medication was much more intense once I was on the medication. Second note on that, the doctors are coming around on it too. So if you're comfortable with your physician, I would highly recommend asking them if you're taking medications, because you don't want to have a bad experience. And then when it comes to taking big doses, I've really had a lot of success with stair stepping, which you can set out a certain amount and just every hour and a half or so, just ease it up. Like you don't have to take one big dose all at once.
Yeah,
You
call that
take
booster pack.
3
Yeah,
small
stair
doses,
step,
like
booster
an
pack,
hour and a
yeah.
half apart. Yeah. Way better. You
I-
can't go back. Once you take it, it's already in there.
But this also echoes what Billy was saying about kind of start and go slow. And there's going to be a gap where science is going to leave us high and dry with no information. And it's got to be this like intuitive thing you figure out yourself. So start slow, do maybe stair step if you're trying to increase dosages. Yeah.
Yeah, so I just wanted to, everything's so positive, which I think is wonderful. Are there any risks?
I feel like the only risk is that it will be an unpleasant, uncomfortable experience, and it will be finite, and it will be over before you know it, and there's lots of unpleasant, uncomfortable experiences in our life, and as Billy mentioned, many of them are there for our growth. So, but there is not like a thing of people having heart attacks on psilocybin or like things like that. I think there is a risk of an unpleasant experience, maybe.
One of the things that Timothy Leary and Ram Dass said about the experience is that set and setting are the most important, two of the most important things. In other words, your mindset and the setting that you take the substance in. If you've got a bad mindset about something tragic just happened to you recently, it's probably not a great time to do a heroic dose. Because things can get very scary. Very scary.
Yeah.
And where do you want to be?
I'm sorry?
What setting would you like?
Oh, what do you do?
Depends on what setting you feel comfortable in. In other words, for me, I like to be outside. I like to be outside and doing things because it's nature and it's all just coming at you. You know what I mean? If you're in a small room and it's dark and you've got your eyes closed, it's all inside your head. And that can be a pretty uncomfortable place to be sometimes. But when you have the world around you to interact with, for me, I prefer that. If you're in a house, I want music on. So there's a number of different things that come to play if the environment changes. That being said, probably the most important thing is your mindset and where you are inside your head.
So I just want to follow up on the set and setting thing. In the clinical studies, they tend to do an indoor thing. There's like a anecdotal concept of if you are out in nature or in music or some sort of external stimulation, then it is like the mushrooms experiencing the human world. And if you are more like quiet and isolated and possibly not, eyes covered in things, then it's that you are exploring the mushroom world. Has people heard that concept before of like, so in the settings in the studies, they tend to cover eyes and bundle in warm cloths, like warm and soft and cozy are feelings that you want in set and setting. In these research settings, right? They do not tend to experiment with things like music stimulation or art stimulation or nature stimulation.
Do they close their eyes?
No.
There are definitely ceremonies that involve darkness. But Tom, you want to follow up on the firm no?
Oh, that's okay. You're going.
No, I please, maybe we're getting.
Well, I think that
Set and setting is a big topic, I guess.
the Mexican culture surrounding psilocybin is pretty open. They may do a ceremony for you. It may be done at night. Might be done around a fire. It might be done with some chanting and the burning of certain objects that create an incense-like situation. Frequently, raw tobacco is used in that situation. But it's also done in the daytime there, and a lot of the sort of the people who went to Mexico to speak to Maria, and there were a lot of people. In fact, the whole industry of sort of psychedelic tourism began with her in Mexico, and so it had opened up a fair amount in terms of the environments that the brujas were doing the magic with, but it was always a ceremony. It's not, and there would be some dietary things that had to be taken into consideration. One of the things about eating, that's generally speaking, you're going to eat fresh mushrooms, which is another whole stack of cats. It's another different environment.
So I feel like I feel like maybe we should take this offline. We all talk about it. We don't want to get too distracted, but I do want to say that the mushroom stones are understood to be for grinding dried mushrooms, and all the chocolate. The mushroom stones are understood by historians, and just a theory, whatever, to be for grinding dried mushrooms. And so many of the recipes that Spanish recorded are with dried mushrooms also. So it's not always fresh.
No.
And there's just so many cultures in Mexico. It's kind of hard to yeah.
And not just Mexico, but lots of different places around the world.
Absolutely.
I'm not speaking from personal experience, but I got so interested in the topic, I've done a ton of reading now and watching all the various documentaries about it. And one thing that comes up over and over again about set and setting is intention, mindset, but it's your intention. And for a lot of things, like if someone's dying of cancer and they're having fear about dying, when they go into a psychedelic experience with a higher dose, they may face things in that experience that are frightening. But you're told over and over again in my reading, what I get told is to surrender. And so one of the ways you could end up perhaps having a negative experience is not knowing that and resisting what's coming.
That's common thing, yeah,
And
that's,
so the surrender seems really important, and that's part of the processing that allows, I think, for the experience to have such a huge impact and to help people overcome the fears of death. But anyway, that hadn't been mentioned, and I just thought I would bring it up because I just encountered it.
I think that is a big part of set and setting, is intention in the letting go versus fighting, and fighting can produce an unpleasant emotions is a commonly expressed thing in the guiding. I think are most people familiar with that concept? Don't fight it, just go where it takes you. Yeah,
There's no way to fight it. Once you're in the river, you move.
yeah.
You can't just jump out, you have to walk out.
If everyone's read these by now, because they've been up there for a minute, but I just would say, here we go, set intentions, check your moods, because besides how much you ate, it's like, are you stressed out? Are you worried about something? You know, like, okay, this one I really like is develop a vocabulary for yourself and for other people you talk to. So we're not going to go around being like, what are the analytical HPLC results on this? And I would like 0.24 milligrams per kilograms and let me get on a scale. That's not going to happen. So we have to have a vocabulary, and that can help you with your journaling and your recording. It can help you put a handle on a complex feeling or an experience, so you can say, that was like this thing, and I have a handle for this complicated set of feelings that I'm trying to achieve or avoid or something, right? So developing the vocabulary is really, really helpful. And then keep a journal and reflect and integrate. But so here's an example of a vocabulary thing I want to share. There's a person who has a thing called Mycozine. It's like a little zine magazine about mycology. And it's my picture, but his idea that is within his friends and social circles, they talk about an experience as being kind of lighthearted, but leaves are dancing. It's not just I'm in a good mood today. Something's kind of happening, And then level 2 is like the trunk of the tree is kind of breathing. If you look close, this is a little more intense on the psychedelic, right? And then tapping roots being like, whoa, it was. So this would be the kind of thing you could say to your cultivator, your purveyor, or to your friend when you're sharing experiences, right? Developing these kind of vocabularies. I have a friend who talks about things being like sparkly and then wavy, and then this, like having a scale that you use for yourself and for sharing with other people is super important. And that's how we do these like building on personal experience and things, is having these kind of vocabularies. Okay, that's the end. And my last thing is a pitch to join Mendocino Coast Mushroom Club if you haven't. Is everyone here members? And then also we have a great website with other stuff. So that's the end. Thank you very much.